Thank you very much. Let me thank David. If I was really smart, I would take a bow, sit down, and live on what David tells you. I want to talk about information theory in biology, which is something that has become really more popular in the last 15 years or so. And I'll talk about a specific gene. It wouldn't be a surprise if it wasn't p53, so it is. And the reason for speaking about p53 is that it's the most commonly mutated gene in human cancers. 55% of all human cancers suffer p53 mutations that inactivate its function. And then another 30% of cancers in humans inactivate the gene itself, or inactivate the protein without a mutation. They do so by modifying the protein with, for example, phosphorylation or methylation. And so the commonality of human cancers is the p53 gene. And then it is otherwise an extremely diverse disorder. It's an disorder of every one of our tissue types and many of our cell types. And it therefore is a challenge in trying to treat it and trying to understand it. But there is this commonality. So I'm going to focus on that commonality tonight. Right? Now, the original title of this talk was that the TP53 gene contains a polymorphic diversity box that controls p53 proteomic plasticity and influences cell fate decisions. There are two reasons I'm changing the talk. The first is, not the most important, but the first is, if you use the word diversity box in your applications to NIH today, so fortunately, everything I'm going to tell you is supported by a private foundation. The second reason is the wonderful collaborator I have, Pierre Hanot. Pierre Hanot is at the University of Grenoble, and he's the head of the cancer center there, and he's an epidemiologist, and he specializes in the epidemiology of p53, both as an inherited disease, but also as a spontaneous disorder. And Pierre makes science fun. We now have a standard mode of doing science together. I usually write a first draft of something that I think is interesting, and he then changes it quite a bit and sends it back. And then he gives me new ideas, so I change the second draft, and the third draft, and the fourth draft. So what I'm going to talk about today, in fact, has been something that has been in the works for about a year and a half. And it's very important that he gets me this next draft before August, because he's French, and he's going to take August off the whole month. And I'm American, and I don't. The new title, is information in the p53 gene. How much information does the p53 gene contain? The p53 gene is a really small part of the human genome. The human genome is 3 times 10 to the 9th base pairs or nucleotides, and the p53 gene is about 20,000 base pairs. Right? And so it's a small part on chromosome 17 on the small arm, right? But it plays a big role, a really big role. And so how much information can you pack into that gene that plays that big role? There's yet a third reason why we use information theory to get at this problem, and that is that as the human genome was being sequenced, this is the year now 2020, 2021, 22, 23, it got better and better with time, right? There were bets on the people who were doing the sequencing, who were working on the genome. There were bets on how many genes there would be in the human genome. After all, we think of ourselves as the most complex of animals, right? Now, we knew at that time that the Drosophila had about 16,000 genes that encoded proteins, and that the worm, the Xenorhabditis, Xenorhabditis elegans, had about 20,000 genes, right? And so the bets were on, and they usually started at 100,000 because we were so much more complex, right? And so when the count was on in the year 2023, for example, everyone was quite surprised to find out we only had 25,000 protein coding genes. Now, we do have other genes which make RNA molecules, and there are about 10,000 of them, and they're repeated often twice or 3 times. So maybe there's 30,000 of them. So maybe there's 55,000 genes in the human genome that makes an organism that we recognize is enormously complex. So where is the information coming from if there's only that many genes for proteins and RNAs that regulate the levels? That's really the question Pierre and I took a year to get to, right? So let me start then by giving you a little history of information theory. We go back to New Jersey. That's the state I work in at the Institute for Advanced Study. There's a mathematician named Claude Shannon who is at the Bell Laboratories in New Jersey. And right around that time, 1948, the World War II has ended. The industry is really starting to boom. And right around that time, telephone calls, direct line telephone calls, were being made for the first time between New York and California. And what Shannon realized was that he could break down those telephone calls. After all, he worked for Bell Labs. He could break down those telephone calls into bits of information that would be added up so that you'd see how complex the problem was. And just think about it. It's tremendously complex. After all, you have a caller who is using language, and the way language is translated into the phone, it then changes to electrons, which go through a copper wire. At that time, didn't go through the air. It went through a copper wire for 3,000 miles. Then the electrons are decoded, right? And then the electrons go back to language, and the person listens, right? And Shannon worked out the mathematics for how much information that would take. And here's where the Institute for Advanced Study comes in. Shannon really was the first one to do quantitative analysis of information theory. And of course, that really was a boon for the computer industry and so forth. So Shannon came and gave a seminar about his information theory at the Institute for Advanced Study. And the mathematicians realized immediately in his equations he had the term P, probability of uncertainty, P log P, probability of uncertainty. Uncertainty means a mistake is made, and it gobbles up the language. Mistakes can be made because the copper wire has a scratch in it, or it heats up too much, or the person calling has made a mistake, and so forth. And those mistakes are random throughout the call. But what's not random is, of course, the language and the electrons and the language that you use at the end. So what Shannon's equation said right away was, why not use 5 copper wires and have the same call 5 times, the random mistakes occur once, and the language itself is reinforced 4 or 5 times, right? And in that way, that's exactly what Bell Labs did, actually. Those wires were 5 and 10 copper wires, right? And the interesting thing was that mathematicians at the Institute for Advanced Study, when they heard his talk, pointed out that P log P, probability of the uncertainty, right, times the log of the uncertainty, was in fact entropy. And that information, therefore, was related to entropy, right? And that if you had I'll put it in the parlance for biologists, but if you had a nucleotide series that was A, right, it's very dull. But now if you had A, B, A, T, C, G, and so forth, and mixes, then the entropy goes up. right? Then the mistakes are made and the entropy goes up, and that gives more information, right? So 1948, we get for the first time a quantitative approach to information theory. Now, it's only 5 years later that Watson and Crick, right? Watson and Crick come along, and they, in fact, build the model for DNA, right? And they make two predictions from the model that is just remarkable predictions. One was, how does the genetic information duplicate itself? And that meant the two helical strands separate, and base pairing, that's really hydrogen bonding between chemicals on each side of the base pairs, the base pairing allowed the duplication to occur. All right? So that was, for the first time, an understanding at the molecular level of how we duplicate our information, not only to build a human being. Human beings have 30 trillion cells. So think of all the duplications just to build the average human being in the room, right? So here we have a situation where duplication is predicted, and it was only 5 or 6 years later, in fact, when Messelson and Stahl proved that the two strands separated, and in fact, DNA was replicated that way, semi-conservative replication. But there's a second thing that they realized, that in 3 times 10 to the 9th nucleotides, the size of the human genome, for example, there was a code. There was A's and T's and G's and C's, only a 4-letter code, 4-letter alphabet, right? And that alphabet, the sequence of that alphabet gave information, right? And therefore, biology for the first time, Darwin never thought about these things. Darwin really settled the situation about how people evolved, but he didn't think about the mechanisms, right? But here now we have replication and we have information, right? And so in the 3 times 10 to the 9th base pairs or nucleotides is information to build a human being, right? A remarkable amount of information, right? And that's why the bet was on that we had, hundreds of thousands of genes and so forth and so on. And people were a little bit surprised and taken aback that we could have only 25 protein coding genes. Proteins are the functional entities of the cell. How does only 25,000 proteins make the complex human beings that we think we are, right? That's the question. And we decided, Pierre and I decided, we try to take a look at that question with the gene p53. There are a couple of reasons for it. One was it's a major player in cancer. But the second reason was we knew a lot about it. If you're a major player in cancer, there's a lot of research in the literature. So I don't want to pretend that I did all this work or Pierre did all this work. We have put it together in what is, I hope you'll see, is an organized fashion. But at the same time, right, we didn't do all the work. It's really the field, right? All right, so let me introduce you to the p53 protein, all right? Now, on the bottom here, you see a box, a linear box, a rectangle, and it has 5 colors in it: red, yellow, green, light blue, and purple, right? And on the top, it numbers the amino acids in the protein. So the nucleotides in the gene make the order of amino acids in the protein. And the protein has a polarity. And at the first, where it starts to be made, it's called the amino terminal end, that's where it says number 1. And it goes all the way to 393 amino acids. So that's the size of this protein, and it's spread out. Now, it doesn't really look like that. It's not a rod. It has the information, first of all, to fold itself up, right? And it folds itself up into 5 domains or units that look like functional and structural units. And in fact, if you separate those 5 units, they'll fold themselves by themselves. They don't need the rest of it to fold. Now that's at the secondary level, and the tertiary level, they'll need the 3rd dimension, all right? And actually what you see here is that the black lines And the number, well, so we number the amino acids. And information comes from where the amino acids are and what they are, right? And so I'm going to just tip you off to the numbers. And it's 1 to 393, that's what position it has, right? And I'll tell you, in fact, that if the first amino acid is arginine, that the letter R stands for arginine. And you can see that all the way on your right-hand side, my left-hand side, at 175. So the amino acid 175 is arginine, right? And it changes to histidine when there's a mutation. And the mechanism, the way in which cancers occur is that they occur by mutation in genes, not only p53, but p53 is one common one, right? All right, and so you can see there are hotspots to the mutations, right? Now let me just take you through very quickly what these domains are. So the red domain is called the transactivation domain, and the reason is that this protein is a transcription factor. Now what that means is that it binds to the DNA, first of all. It sees a sequence in the DNA, it sees actually 20 base pairs, and that's 2 turns of the helix, and it will bind to that sequence. And the sequence happens to be degenerate, so it binds to many sequences that have that subsequence, degenerate sequence, right? And then it promotes the messenger RNA being made, copying the DNA, so the messenger RNA can then be translated into a protein, right? And so here we have a situation where the transactivation domain is the place where RNA polymerase, so this protein has bound, the green is going to be the DNA binding domain, bound to the DNA, and now RNA polymerase, the enzyme that copies RNA from DNA, actually is bound to 2 spots in the transactivation domain. And those 2 spots give rise to 2 different sets of genes that are made, and there may be 300 genes that we know of so far that p53 transcribes. So that means out of the 25,000 genes I told you about, or 55,000 genes I told you about, it transcribes about 300. And that's what transcription factors do. They give you the specificity of information that's going to be in that cell type, right? Then the yellow spot is called the proline domain because it has a sequence that's very commonly has proline. Proline, any amino acid proline. And that sequence is a known protein-protein interactor, right? So here we have RNA polymerase binding in the red domain. In the yellow domain, a large number of other proteins communicating with p53 to the rest of the cell in a protein-protein interaction, right? Then the green is the DNA binding domain, and you'll notice right away that the great majority of mutations are in the DNA binding domain, and what they do is they inactivate the ability of the protein to bind to DNA, and it no longer is a transcription factor, and the loss of that those transcripts, the loss of that messenger RNAs from the gene, the loss of the information, right, is what's giving rise to the first steps in cancer. Then in light blue, there is an interesting domain which takes one of the proteins, this is just one of the proteins, p53, right, and it makes a tetramer of it, right? So it does so by making a dimer of a dimer. And so actually the transcription factor is a tetramer, but it's a homotetramer. It's the same molecules again and again and again 4 times. And that blue domain has a few mutations in it, and that will stop the transcription as well. And it's a tetramer, and is one of the things when I told you it binds to 20 base pairs, that most proteins will see 5 base pairs. So if you're a tetramer, now you can see 20, right? And so it gives you a bigger range to play with. And finally, the purple domain is called the regulatory domain. And the reason it's called the regulatory domain is if you modify the protein with phosphorylation or methylation or acetylation, you regulate the activity of the protein, all right? So you've just been introduced in a pretty detailed way to information that the protein holds, right? Okay, now function, right? This, of course, is the real action here. p53 is a stress responder. Now, what's quite remarkable is it responds to many stresses. If I say stress, I bet in the audience, you'll probably think of 5 or 10 different things called stress, right? I mean, it could be psychological stress, and it does. It responds to psychological stress. Chronic stress, psychologically, p53 will respond to, right? But the most common thing it responds to is damage to the DNA, because damage to the DNA causes mutations or death, right? And we damage our DNA every day, right? Coming through the walls of the hotel right now is radiation which actually goes through us and can break the DNA. In other words, the 3 times 10 to the 9th base pairs could be broken, right? That's one kind of damage. There are actually 10 or 12 different kinds of damage. Chemical damage, chemical carcinogens. So what we ate tonight, I guarantee you it looked really healthy, so it wouldn't be a problem. But, but nonetheless, what we eat is what we breathe is some of the problems, right? We are repairing ourselves all the time, right? And there are many mutations made every day. And there are 30 trillion cells in which those mutations can be made. So as soon as these stresses occur, p53 sees the stress. It doesn't see the stress itself. It has a sensor, which in this case, now we'll get into jargon and I won't bother you with it, but the stressor happens to be called ATM. It's a gene which will cause leukemias. So it is in the pathway of cancers. And it binds to single and double-stranded breaks in the DNA. It taps a mediator, which is a protein kinase, which is called CHEK2, and that amplifies the signal. So we're using enzymes to amplify the signal, and then that phosphorylation of p53 will activate it, so now it's a transcription factor. But it inactivates another protein bound to p53 called MDM2. For those cognoscenti in the audience, MDM2 is a ubiquitin ligase. That means it puts a little peptide on the p53 protein, and when you have enough of them, it shuttles the p53 protein into a proteasome, which degrades it. That means that p53 is regulated at the post-translational level, after the protein's made, right? And in fact, in T cells, that's one of the cells in the immune system, right? The half-life of p53 in a cell is 6 minutes. And that's because it's made and degraded, right? It's being kept in check. It's potentially dangerous, as you'll see. It's being kept in check, right? But in fibroblasts, it's 20 minutes. Right? Now, there's a second reason why this strategy has been approached, and that is that if you wanted to do something, if a stress occurred and you had to transcribe a gene, that'll take hours. This takes minutes, right? So this stress responder is your stress responder, which in 5 minutes can get half the concentration doubled, right? All right, so p53 and MDM2 form their own little box, and they regulate each other. Why? Because p53 transcribes the MDM2 gene. So just think about that. You have a gene that transcribes its own negative regulator, right? That's exactly like, that's like foxes and hares, right, in the environment. If the foxes go up, the hares go down, then the foxes go down, the hares go up, right? And you get an oscillator. And we see that in cells. p53 oscillates, all right? So you can see from the whole structure here, right? Now, what does it do? The first thing it does at this point is it makes a decision. And that's a very interesting thing to say about information theory. It makes a decision, right? One path goes to death, right? And there are 5 ways to kill a cell. These are programmed cell deaths. You have in your genes a set of informations to kill off cells that you have. And in fact, you do this every day, right? Your cells are turning over every day. The half-life of the cells in your gut are 4 days. The half-life in the blood is 28 days. The half-life in skin is about 36 days, right? So there's a turnover. And p53, if it sees damage it will kill. And there's 5 ways to kill. Now, for a long time, we didn't know why there would be 5 ways to kill. I mean, this isn't one enough, right? However, what we now start to understand is that with death comes the ability to communicate with the immune system and immunize against that bad thing that happened that made death, right? And so cancer has genes in it that are immunizing, right, with death, right? p53 will stop the cancer and actually immunize against those genes that are causing cancer, right? And that's the whole immunotherapy revolution, and this is a player in the revolution, right? Okay, but there's an alternative, a happy alternative, right? Which is repair, right? I mean, why? If you're not going to die, you should repair. And so there's cell cycle arrest, there's no more division. There's repair processes, and p53 regulates the genes for repair, and then there's autoregulation to turn the cell cycle back on. And so this decision is made by specific amino acid in the p53 gene protein, right? In the p53 protein. And we'll start to get, and that's information. We'll start to get to that now. All right. Now, I'm going to introduce you to something that's conceptually very simple, but puzzled people for a long time why we do it, right? And that's splicing, right? So why it's conceptually very simple is we know about how movies are put together. They're spliced together. You know, all the parts are made, and then they're spliced together, and they look better that way, right? Well, that happens with our genes too. And so I told you that the p53 gene was 20,000 base pairs, right? But all you now have to do is take 393 times 3, that's the number of A's, T's, G's, and C's that will give rise to an amino acid, times 3, and you'll see that there's very little part of the gene that's made the protein, and there's a large part of the gene that actually is called the intron and doesn't ever go to the protein because it's spliced out, right? Now, it turns out there are 12 different splice forms of p53, and they form a pattern that really is very informative. And so you can see the pattern here. So let me tell you that the colorful, well, the first line under protein, you see the gene, and you see it has, wherever you see a box, it has what's called an exon, which is coding for an amino acid or having the messenger RNA in the position to make the amino acid, right? So TP53 is the full-length protein right at the top, and it has, as I told you, the transactivation domain, it's green here. The proline domain, it's white here. It has the DNA binding domain, it's yellow here. And then it has a full length called alpha, which is an orange color. And that's the full-length gene, right? But if you delete the first 40 amino acids, right, because there's an A, U, G in 40 amino acids, there's a start codon in, I have to remember that some people don't know AUG is a start codon, right? There's a start codon, meaning I'll start here, right, at amino acid 40. And so it truncates the transactivation domain, and if you're looking for information here, it stunts, remember I said there's 2 transactivation sites, it gets rid of one and keeps the other, all right? So that's called delta 40. Now the language is easy right here. Delta 40 p53, and it picks up alpha, beta, or gamma, which are 3 different carboxy terminus, or the other end of the proteins, right? There's a delta 133. 40 and 133 we'll dwell on for a few minutes, and then there's a delta 160. We don't actually know what 160 does yet, all right? So there are 12 different splice forms. So that has taken the information up 12-fold, because these splice forms are going to, you'll see, have different functions than the whole full length, right? So that's already a bit of information theory here. All right, now I'm going to tell you what the functions are. For delta 40, it lacks the first transactivation domain, which I can tell you is at 25 and 26. And the first transactivation domain was also the binding site for the ubiquitin ligase MDM2, which degraded the protein. So that means delta 40 is more stable than is the other protein, right? The strategies are starting to mount up here in interesting ways, right? The isoform is produced either by an internal AUG, right? That means that the ribosomes get on the messenger RNA at 40, right? And to start making a methionine, right? Or sometimes it's spliced. Now what are the functions? And this is a remarkable thing that we were, really, all of us were unprepared. It's due to a wonderful woman named Heidi Scrabel who actually figured this out. The most highly expressed p53 isoform, an isoform is one of the 12 proteins. The most highly expressed of the 12 proteins in embryonic stem cells in the mouse, we can't do this with humans, but in the mouse is delta 40. That means it's involved in development. In the mouse, the first step is you have a fertilized egg. It grows to thousands of cells. It makes a blastocyst, which is just a sphere, right? And then it has a few cells that are going to make the mouse. Those cells make delta 40 more than any other transcript of p53. Now, what is its functions in the embryo? Haploinsufficiency, that means only one copy of delta 40. Usually there'd be two copies because we're diploid. We have two sets of chromosomes. So if there's one copy of delta 40, there's a loss of these stem cells. That means delta 40 immortalizes stem cells and keeps them alive in the embryo, right? And you lose it if you don't have it, or if you have too little of it, right? Now instead of one copy, you do 3 copies, right? And now you have no stem cells again because all the cells have differentiated. Stem cells go to differentiated cells, and that's what it's forced it to do. So delta 40 is something to do with stem cells. Stem cells are the cells that are the targets for mutations in cancer. Right? So just the information that's piling up is starting, right? Okay, so that's delta 40. And the next guy is quite a surprise for everybody as well. Delta 133, right? That is missing the first 133 amino acids, missing the proline domain, missing the transactivation domain. This guy shouldn't be able to transcribe, right? It has just the DNA binding domain. In fact, we thought it was at first when we found it a repressor. Why? Because it binds to DNA, but it doesn't transcribe, right? So we thought it would be a negative regulator. It's not. It's a positive regulator, right? So what is it about? First of all, if you take delta 133 alpha, put it in a virus, take the virus, infect the human cell with it, it makes the cell divide. It causes DNA repair. It causes telomere, that's the DNA at the ends of the chromosomes, to be made properly. It has tremendous mitochondrial activity, right? This is turning on everything. Literally, instead of being what p53 is, a tumor suppressor gene, it's acting like an oncogene. It's forcing cell division. And Carl June, a name that many of you who are in the audience that were biologists will know, worked on getting T cells, the killer cells, to kill the cancer cells with a proper receptor. And the only thing that stopped them was that after there's killing a few times, the cells undergo senescence or exhaustion. Now, that's a good thing, even though it sounds like a terrible thing, because if they kept growing on, that would cause a leukemia or a lymphoma, right? If you take exhausted cells and put delta 133 in it, this is called June's experiment, put 133 in it, it will cause the exhausted cells to now activate and kill cancer cells. So we thought right away that was terrific because now you don't have to worry about exhausted cells. But of course you do. This is growing on ever more. And there could be a leukemia as a result. He's done all his experiments in mice so far, but we'll see. We'll see. All right. So we've suddenly, out of the splicing components, gotten totally new information about the p53 gene, some of it completely contradictory to what we see the full-length gene do. All right, now I'm going to the second thing that happens to any gene. One of the virtues of this talk is this is as much I could have picked any gene and you would have seen splicing and it would have done different things, and now you would have seen what are called polymorphisms. Those are mutations that occur over evolutionary time frames that if they improve the protein, they'll be kept, and if they destroy the protein, it'll be lost, and if they do nothing, could be kept or could be lost, right? All right, so here's the gene on top, right? That's the exons and the introns. There's the exon-intron structure, right? And there are the polymorphisms. First of all, they're all in the middle of the gene, the polymorphisms. Look at the red ones are in exons, the blue ones are in introns, right? That's the diversity box. Why? Because, so I'll take the example I'll use from now on to show you a good example of what this could do. The R72P, right? That's an E4 exon, right? R72P, that's arginine, codon 72 to proline, right? Okay. That guy is in homozygous, it's homozygous p53. It's going to be in everybody in p53 who's born around the equator, and then everybody born in Scandinavia, it's going to be arginine. And what we're talking about here is a gradient between the equator and the North Pole, right, that this guy has picked up as different environments, and it's arginine at the North Pole and proline at the equator, right? Now, one of the things that was obvious from the first is you get direct sunlight at the equator, you get indirect sunlight, and that's in the proline domain, where skin cancers occur. So it starts to make sense. But now that I've said everybody at the equator has proline, everybody in Scandinavia has arginine, I'm now speaking about ancestry, right? I'm now speaking for the first time about ethnic groups and ancestry, right? And the cancers of Africans and the cancers of Europeans are different. And here's something that's different all across these polymorphisms, right? And the reason that's so important is just think about how drugs are made, right? Drugs are made in Europe and the United States, and they're tested on Caucasians, and there may be 5% of the population black that's tested them, and maybe 6%, 7% Asian, and that's testing them, and yet they all have different polymorphisms. And here's an example of polymorphisms in a gene which is intimately involved in cancer, and we know only how the drugs work in Caucasians or European ancestry, right? So let me just work the ancestry for you. It's the primates that gave rise to mostly bonobos and what are they called? Chimpanzees gave rise to humans, right? Happened about 300,000 years ago, and it happened in southern Africa. Now we know that pretty surely both by the bones that we find, but also by the DNA we find in those bones, right? And so 300,000 years ago, humans are anatomically, right, human. However, they're not quite human in terms of cultural things. They use primitive tools. They have problems with, they don't draw on caves, right? That only happens about 200,000 years ago. And then they're for the first times in the caves in Israel. And then about 50,000 or 100,000 years ago, humans are in the caves in Spain and all around the Mediterranean. A second group goes down into India and Australia, right? A third group goes north to China and China. What wasn't China then, of course, but whatever it was, right? So, and that's when these things started changing. And they started changing because the environment was changing, right? Now, let's talk about function of these polymorphisms, because they are giving information to the gene, right? All right, so here's an example of just how many different combinations of p53 you can make when you put the splicing together and the polymorphisms together, right? The isoform variant architecture, right? If you have alpha, beta, and gamma as the isoforms, right, at the C-terminus, right? You have two isoforms for the R72P, right? That's the one I told you is very different in Africans and African ancestry and Caucasian ancestry. And the same thing is true with delta 40. And delta 133 has one isoform because it only uses beta and gamma and it doesn't have the tetramerization domain, right? And 160 uses one isoform, right? The oligomerization then gives you the possibility that a full-length p53 works with 40 and 133, right? And that's what allows it to be a transcription factor. And that's what allows it to have a phenotype, right? So when you add this all up, what you have is n is the total number of isoforms. Here we have n is the total number of isoforms. Alpha is the total number of isoforms in alpha, in the alpha C-terminus. And if you're a monomer, a dimer, or a trimer, or a tetramer rather, of p53, you're in that group. And that's n plus 1, and n plus 3. And it's the factorial 2 and factorial 4. And it turns out there's 315 different proteins you can make, or combinations of proteins you can make with the isoforms and the splicing sites, right? So that's really multiplying information in an evolutionary way. Just think of writing code now. I mean, I'll bet everybody under a certain age here writes code, right? And everybody over a certain age doesn't. So I would like to tell you, even the immune systems are better in those who write code. So what we have here is a situation of writing code, the evolutionary code, right? Having 25,000 genes, but if each of these genes had 315 combinations, think of what the multiplies are, right? All right. Now it turns out that's not true. And it's not true because of another biological principle I promise I'll introduce you to. Lots of biological principles. And that's called linkage disequilibrium. You have to be a geneticist to really love linkage disequilibrium. What it says is that when two chromosomes come together and they pair, right, they undergo recombination. So what happens is you get a little bit of the first chromosome, it crosses over to the second chromosome, it copies the second chromosome, and then you get a hybrid chromosome, right? That would take polymorphisms and make them diverse, right? But we didn't see them as diverse. We saw them as either being in Africa or in Scandinavia. So we said something is wrong here because in Africa in particular, there would be lots of recombination that have occurred over 300,000 years. But also between African Americans and Caucasian Americans, there's lots of recombination that has occurred in the mixtures, right? And so we started looking for the failure of recombination to occur. That's called linkage disequilibrium. You're throwing the equilibrium out. And sure enough, the polymorphisms in red at the N terminus, that's that's a G to a C or an R72P, pair up with only one polymorphism at the C terminus, right, at the other end. And those in green pair up with the other two. So that means there's something about combinations that are not viable. That if they're viable, they don't reproduce well, right? Actually, David and I just discovered, while we're at the meeting here, a situation where p53 is inherited, sometimes you get this kind of change in sex ratio. And change it not only in sex ratio, but changes in the transmission of the gene itself, right? That's this linkage disequilibrium at work. So that limits, that takes 315 down to some number, and that means we have to go through the populations, Asian, African, and European, right? Go through, and actually South American, because there's quite a mixture in South America, right? And ask what's the protein sequences, and which ones occur and which ones don't. And so far, we have 5 clear examples of patterns, right? All right. Now, I'm just going to finish up. I may be overtime, but am I overtime, David? A little bit. A little bit, yeah. I'm just going to tell you the differences in the cancers that are happening in Africa versus in Europe, right? And look at number 2. That's the ones that are about your bloodstream, right? A multiple myeloma is a B-cell cancer, right? It actually has an inherited component and a spontaneous component, and p53 is involved in both. A polymorphism is involved in the inherited component, and a mutation, a spontaneous mutation, is involved in the other, right? And Africans, so if you have a population of mixed African ancestry and European ancestry, let's say you have 100,000 people, 66% of the African population, multiple myeloma will be found in Africans 66% of the time, and in Europeans 34% of the time. That means the frequency of the disease is different. The frequency of the cancer is different, right? That's probably due to the concentration of p53, right? And the same thing is true with the other half of the immune system, the T cell system. In Africans, 60% in T cells and 40% in Europeans. And then the one that really blows my mind is it's the exact opposite. In a tumor of testicular teratocarcinomas, that's a tumor of the stem cells of the testicles, right? African-European ratio is 1 to 33. That's inverted, right? 1 African for 33 Caucasians, right? Europeans. So there's something about this molecule that's giving rise to frequency differences, and we see that all along the process. I'm just going to show you, I'm going to skip a couple of things because, yeah, so this is a good example. That we did this, so once we realized what we were onto, about 3/4 of the way through the year, we hooked up with a wonderful guy named John Crofton, who's the head of City of Hope Cancer Center. And he had been sequencing African Americans and Caucasian Americans, European Americans, and he has looked at them as European ancestry and whether you have the wild type p53 or the mutant p53, right? You can see that every point is an example, and the plot is European ancestry. The reason we can say that, so let me just say, in most experiments, everybody is self-proclaiming what they are. So that may be mixtures and so forth and so on. But we could look at the genes because we're sequencing the whole genome, and we know that some are African and some are of African origin and some are of European origin. So that's what that plots. 100% Caucasian, you have wild type, right? And here you have Africans, there are 20 to 30% Caucasians in American populations because of all the inbreeding that had, all the outbreeding that had occurred for years and years over in America in the slave trade. There's only 2 mutations in that group for p53 in the Africans, and there's lots of mutations for p53 in the Caucasian ancestry. And then that means something is shown on the next, the slide over, right? Where the red is if you've lost both of the copies of p53 for mutation, you actually survive less well than you do if you don't have that loss, right? So it has function, and it's both frequency and survival. That's an important set of functions here. All right, so look at the information grow. That's the point I want to make. We really sort of nailed this now by, we just resequenced a number of individuals here and extended the numbers, and we're just analyzing the data now. But This is really looking at biology in a little bit different way. I mean, the biologists here, I think, will tell you that people don't analyze the gene and its isoforms and its polymorphisms and its functions in this kind of detail to look for information. And it's information that's important for disease processes. It's information that's important for evolutionary biology. It's information that really gives one a whole sense of what the biological revolution has been about in the last 30 years. Thank you. But I'm happy to answer some questions. Yes?
So,
Wait.
okay.
You need a mic.
I was going to speak very loudly. I'm Alfred Spector. A wonderful presentation, not that I understood all of it, but I have a feeling some of us may not have grokked everything you said, but it's, we got the gist. You make a compelling case that there'll be very big differences in different subpopulations.
Yeah.
So that's clear, and you have a compelling argument that's plausible, at least, that you have a reason for it happening and such. So that's all very good. What are the implications, though, on therapy?
Yeah.
So if we have all these isomorphisms
Yeah.
and polymorphisms and all these differences, does that mean that we have the complexity then of whatever the combinatorics and the number of different drugs or treatments that we
Yeah.
need to have? Or is somehow this going to collapse
So
into
I can
some
tell
more
you really good.
beneficial way?
I'm going to tell you two good stories about therapy. Just the sociology of the pharmaceutical industry is such that the pharmaceutical companies, once they get some good drugs in a particular kind of cancer, they focus on it. They make more. They corner the market. Right. So I happen to be involved with one of them. Janssen is involved with multiple myeloma, just the thing I've been sequencing. And of course I keep them informed, and right, because your question really is exactly what they didn't achieve at the first round through. Their numbers were 95% Caucasians or Europeans, right, and about 5% Africans. And of course, what they do is, for the FDA, you give the statistics of everything, right? Every person is important, and you lump them together. And of course, you know nothing about the African ancestry, and you know only really about the European ancestry. So the FDA, of course, caught on, and then insisted, yes, we'll license the drug. It's a very good drug. It's called daratumumab. And it really helps people have lifespan. It doesn't cure, but it really gives extensive lifespan. And so they went ahead and went to a number of places where African Americans but now notice in the population, there are more African Americans that get it than Europeans. And yet they were less involved in the clinical trials, right? So the sociology of this is yet another part of the questions that comes up, right? And I would say it's a little early so they really got a good number of African Americans. Now, in fact, I'm going to go backwards because it'll illustrate, if I can, yeah, I can. This illustrates, look at the European ancestry and look at the genetics of individuals that have been in the United States from Africa for the last 300 years or more, right, is really mixed. It's a real mixture, right? And that's a complication in itself. And we have to read the polymorphisms of the p53. And there certainly that isn't certainly the only gene. I'm sure there are others, right? Okay, so that's been one problem that I'll use as a qualification of the data. But there was a great irony in the early data. The early data, which is just published and released, is that the daratumumab actually worked better in the Africans than it did in Caucasians. And that the FDA held up the drug longer because the Caucasians were less, they were the majority, but the longer group, right? So it's crazy, but the truth is when you have the knowledge and you want to go for it, then it makes some sense to go for it. And talk about diversity boxes. This is really the end of it. I want to show one other thing, which is really a really remarkable surprise. It's this, all right? This is a really bad cancer. Non-small cell lung cancer is the single largest cancer in the world. And that's really because of smoking. Right? And it's still the largest cancer in the United States, in spite of the fact that very few people in this room will smoke, right? So remember I told you that at codon 72, you're either proline, PP, or arginine, RR, or you could be the heterozygote, right? Someone who had one chromosome that's P, one that's R, right? And then we asked the question, which of these had p53 mutations? And look at this. If you're proline, that means if you're from an African ancestry, but originally, now it's European, because the truth of the matter is, in Poland and in Italy, in southern France, proline is a presence in heterozygous state. Right? So if you're proline, you have 65% of the cases had p53 mutations. If you're arginine, it was only 40%. And here the heterozygote's in the middle, right? That says two things that are really remarkable, right? First of all, here's a gene whose polymorphisms are allowing it to inform its own mutation rate. This is a spontaneous mutation rate. Now, the only hypothesis we have for that is that it's at higher levels in one of the groups than the other. And we'll check that. We can test that now in the populations, right? But it says that genes can regulate their own mutation rates. It's typically biology, right? It's not like Einstein's laws, right? Where you can go to the 9th decimal place. So the p-value is 0.01. So I'm sure that the cosmologists among us would not have accepted this. But this is really was a remarkable thing that came out, because we could ask the questions for the first time about what this does. Yes.
Hi. I really enjoyed your presentation. I actually work with embryonic stem cells, so I found the vignette in the middle actually very interesting. So if I understand, when you delete p53 in a mouse, it goes up fine to become an adult, right? And it develops cancer at some point.
Yeah.
But when you claim that this 2010 Genes in Development paper, that this delta 40 isoform is required for embryonic stem cells. So I guess those two seem to be in contradiction.
Yeah.
So teach me. I'd love to learn.
What Heidi Scrabble did in her papers was the following. First of all, the reason why, this is a very good example of just reinterpreting a result gives you faith in somebody. When Heidi Scrabble published that result I showed you, people didn't believe it. And the reason is that if you knock out p53, that means there's no gene there. You can't get a delta 40 or anything else. The mouse lives. The mouse is born.
Yeah.
Right?
Right.
All right.
Yeah. Mm-hmm.
Now, what I've just said is that the mouse doesn't if it's haploid for 40 and triploid for 40, right? That means it's the ratio of the wild type transcript to the 40, right? That's important. As long as you now know that these two facts are true, right? We thought it was a non-essential gene. We thought it was, when anybody asks me for the short answer of what p53 is, I say p53 is a tumor suppressor. It enforces fidelity by death, right? Because the mistakes are made by the damaged DNA and so forth and so on. So it's not essential. It's enforcing fidelity by death and giving us a good quality of life. It gives us longer lifespan. It's selected for because that means we have more babies. Right? But the way that Heidi Scrabble did her experiments was she was putting in delta 40 with a wild type, and the ratio was changing when you made a triploid and when you made a haploid. Right?
Has someone deleted the native delta 40?
Yeah,
not
yeah.
by overexpression?
Yeah. When you delete delta 40, you get cancer. And
Okay.
that's because the first transactivation domain makes P21, which is the protein that stops the cell cycle and so forth.
Right.
So unless you get death, that's what you need to get away from cancer. So some mice get cancers and things like that. So it's not so simple as just deleting the first 40 amino acids.
I see. Okay, thank you.
Hi. Thank you so much for the lecture. I feel like I would be almost like committing some type of mortal sin by asking you about a non-cancer disease,
Yes,
but I'm
it's
gonna
okay.
I'm gonna do it anyway. So I feel like cancer was one of the most interesting diseases for like biomajors to look at, because it's very clear that it has a genetic basis and that genetic dysregulations cause it. However, I think there's something really interesting about the way you've been looking at biology, about like how information has been a driver for everything. The thing is that if you look at a lot of neurodegenerative diseases, a lot of metabolic diseases, a lot of fibrotic diseases, you just don't find that genetic predisposition. I'm wondering, what is that like missing source of information?
Yes,
or
I'm going to give you what I would say is still in strong hypothesis form, right? And you're going to have to forgive me because I'm going to forget the name. I have forgotten, that's why I'm going to ask for forgiveness, the name of the woman who did the work, right? But I can tell you, she's a neurologist and she's at UC San Francisco, and they have a whole neurology department where they look at neurological diseases that are mediated by the immune system, right? And they are sex-determined diseases. In other words, the neurological diseases occur in women much more than men, right? All that's true, right? And you'd say, gee, that's not determined by genes, right? But it is. And the reason is they really have found out a very plausible explanation for these disorders. And the explanation is the inactive X chromosome. So now I have to give another lecture, but I don't mind. So women are mosaics, and they're very interesting mosaics, right? And men are not mosaics here. But women are interesting because they have two X's, men have one X, and dosage is important. So women inactivate one of their X's. And they do that with a little RNA. Remember I said there are RNA genes that regulate things? That RNA is called exist. And exist binds to randomly binds to one of their X chromosomes and inactivates it. By inactivating, I mean no genes on that X chromosome are expressed. It's not entirely true. Some genes are expressed, but most genes are not expressed, right? Okay. All right. So that was discovered years ago, and really beautiful set of experiments, first in mice, then in humans. That's why, for instance, factor VIII hemophilia is a very interesting disease where women are carriers of it, right? It's a disease that men get much more than women, but that's because women are mosaics of the inactive X, and that gene for hemophilia A is on the X chromosome, right? So women always have a backup because they're mosaics. So even though half their cells aren't producing factor VIII, that it doesn't mean anything because the other half of the cells rescue them, right? So now let me get back to the neurological cases. What this woman found was, now I'm going to lose a ton of people, but the biologists will be happy. So, all right. So the inactive X inactivates, right? But in women, in different women, it inactivates a different degree of gene inactivation. And the neurologists have shown this, that in some women, the ratio of the amount of enzyme they're making from a gene on the X chromosome is 1.5, not 1. Men make 1 because they have one X, right? Women make 1 because they inactivate the X. But in some women, they don't inactivate the X. They inactivate most of it, but not all of it, right? So they try to track down these genes that are leaky. That's what the expression would be, leaky in biology, because the gene is just dribbling through, and suddenly you're making more of something, right? That's why biology is so wonderful. It's more of something can be bad, right? And what it is that they're making is the gene that's really leaky is a trimethyltransferase, a histone trimethyltransferase. Now, the histone trimethyltransferases regulate genes. They regulate genes epigenetically, not genetically. They regulate it after the fact, right? And they do so by putting trimethyl groups on histones, which are the proteins that package the chromosomes. So that means you could express the chromosome in a leaky fashion in some women, right? And everybody else is, the men just have one X, so they're not expressing it, right? Because they got their X working, right? And most women who have an activated X efficiently, right? So that explains a couple of things. Why it's sex limited, right? That's autoimmune diseases are sex limited and neurological, right? And in this case, it was a neurological phenotype. Now, I don't remember the name of this woman, but I tell you what, I wish I was, I try to be, there's a very fair criticism of my whole career, I have to say. And that is, I focused on one gene. That sounds like a little bit narrow, right? So I feel defensive about that. And so I go to lectures that are about the brain is very interesting. And I taught immunology at Princeton, for example. I mean, I taught it only to learn immunology so that I wasn't just focusing on one gene, right? So it was the Simons Foundation, it was a private foundation, who supported this group at UC San Francisco. And the Simons Foundation, she gave a lecture at the Simons. I go up to New York to go to that foundation's lectures. And it was remarkable because it helps explain a lot of neurological disorders that are sex limited, that are immune mediated. Yes.
Question over here.
Oh.
Yeah, hello. Thank you for the lecture. Yep, over here.
Yeah.
Yeah, so I was quite interested by the discussion you had somewhat early on about the combinatorial possibilities with the different isoforms of p53 and their dimerization and tetramerization. So I'm kind of, this is from the perspective of a chemist, so please forgive me if I'm misunderstanding, but so I was thinking that, in a cell you have these alpha, beta, gamma isoforms of p53, and they have some equilibrium balance of...
Two of them don't make tetramers.
Right, so they have some equilibrium balance of tetramers and dimers.
Exactly, because...
So I was wondering if there's any work or how we might go about thinking about how to sort of disentangle the effects of the presence of certain polymorphs themselves versus their effects on this equilibrium balance of tetramerization versus dimerization.
I'm sure there is, but we really haven't even started trying to get at that. There's two ways to go about it. You could either use PCR or antibodies. Once you have splice forms, it's missing something. So the ratio of the N terminus to the C terminus changes, right? And you could pick that up by PCR or digital PCR, or you could pick it up with monoclonal antibodies. So, but it means settling down, it means having a graduate student that's going to make all the reagents and then do all the experiments.
Time for one more question. And I'm going to go way up here.
Thanks. Thanks for the really interesting talk. So circling back to the question of therapy, one of the questions that was really kind of potent in my mind is, if you have so much diversity, how do you account for that in the clinical trial, given that the clinical trial is just so darn expensive, right? Like it's almost like limitingly expensive. Like, have you thought about what kind of paradigm shift we might have to be able to achieve more personalized medicine? Because we're just not set up for that right now.
There's someone who's here right behind you who's thought about this a lot more than I have. So you want to answer that question? We'll get a little diversity. We're going to get a little diversity in here.
So some of you will know I really shouldn't be answering this question. Bill Dowley, for example, probably knows I shouldn't be answering it. But one of the things, though, is, and you brought it up earlier, is how do you optimize getting the right subpopulations in the trial and knowing who they are with taking into account the cost of the trial in that. So the example you gave is if you use the wrong population, which in this case turned out to be people of non-African descent,
that's right,
you had to do a much larger trial to get statistical significance.
Exactly.
And that was unfortunate. On the other hand, if you try to have to do every possible subpopulation in the trial, that's also very, very expensive. And that'll delay the results in getting the trial. So one of the things that I think is going on is if you have some hypotheses as to where the drug will be efficacious in advance, you can, and perhaps you can develop those with some sophisticated machine learning techniques now, then you can do a better job of optimizing the enrollment in the trial and doing stochastically a better job of balancing competing objectives.
I'll also point out the complication of the fact that we are all interbreeding, and defining groups becomes a very complicated problem. And if you start doing things like we did in this study, using people's polymorphisms who happen to reside in Africa and people who happen to reside in Europe, right, you can get it wrong, right? I mean, Africa was colonized by Europeans for 100 years, and everywhere you go. I mean, that's it really is complicated. And so classification in this sense, I mean, genetics is meant to be really expansive. I mean, try every combination. And, that's different than finding the drug that works on some people but not other people. And it's a very complicated social issue. You see this, I have to say, this was good for my education. When we started doing, we realized that African Americans in multiple myeloma, for example, had polymorphisms that you could identify that would allow you to test against the drug, and that Europeans' ancestry would allow you to test against the drug. And you could stratify just by those polymorphisms, and you wouldn't get all of them, but you'd get some subset of them, things, right? So I went to my first meeting where disparities of giving medicines to people in the United States were being discussed. And more than half of the work is in social sciences. And that's fair, because the ability to get medical care, the ability to get insurance, the ability to be in the right place at the right time, to be enrolled in a clinical trial, all these things are honest. Socioeconomic disparities, they're all honest things, right? And the guy so I was there with mostly social scientists who were beating the drum, which were inferred anti-genetics, right? That it was racial disparities that was important. It wasn't genetics, right? And yet I was to give this talk that I gave you about the genetics, right? And the guy before me was a wonderful man. I'd gotten to know him quite well. He's a social scientist, and he's in New Orleans, and he's a wonderful speaker, right? And he went and he gave his talk proving that genetics was not important, right? So fortunately there was a coffee break, and I went up to him and I said, you know, he's African American, this guy, and I'm not. And so that was the complication of being in a meeting that had, I never was in a meeting that had that as issues, right? So I went up to him and I told him what I would say, right? And he looked at me, he forgave me, so to speak, right? What he said to me was, you are smart enough to come and tell me what you're going to do, and I'm smart enough to tell you it's good, right? And so that's when I learned communication there's a good lesson to that about communication, because I thought I'd get killed. I thought there would be, 7 reasons why, and there are reasons. I mean, you could see the African American group that we looked at are very diverse, and how to classify that group is, difficult to say the least. So. Science is a wonderful thing because it gets you into all sorts of places you never think you're going. Thank you very much.